And also, let me draw you another mental map. So we've worked through all these. We worked through the whole organ, this muscle, and then this bundle of muscle cells called a fascicle going into a single muscle cell called a myofiber. Now we're looking at these structural components, this modified cytoskeleton called a myofibril. We know that they're composed of multiple sarcomeres, and these sarcomeres are composed of these myofilaments, both thick and thin. Our thick myofilament is called myosin, and our thin myofilament is mostly made of actin. So now I'm going to draw a sarcomere, and we're going to keep working through this stuff. Alright, cool. So we are looking at one sarcomere right now, and myofibrils are made up of hundreds of these. So let's use dark gray to label some stuff. The first thing that we want to label are the ends of these sarcomeres. These are called Z lines. We can also call them Z discs. Either one. We know that we have these thin myofilaments, and these are mostly made of actin. We have these thick myofilaments. These are made of myosin. We have the purple over there. That's going to be, this is an elastic myofilament. We don't talk about it too much, but it's what's connecting these thick myofilaments to the Z lines or Z discs. This is called titin. And I don't think he said it this year, but he said it in past years. The protein that makes up the Z line is called nebulin. So whenever you get a nebulizing treatment for an asthmatic attack, I think it's relaxing this nebulin. Okay, is there anything else that we want to add before? Okay, so talking about these different regions of this sarcomere, and I do certain colors for specific reasons, so I'll keep these colors consistent throughout this lecture. So if you're following along with color, I try to do the same colors I do. If not, create a system that works for you and stick to it. And I'll get rid of this. And this. Alright, so we can do this one of two ways, and we'll do both. You can just decide what works for you. You have to find the I band. You have to find the A band. And you have to find the H zone. The other ways that you can do this, you can say that you need to find where you only have thin myofilaments, where you only thin. Where do you only have thick myofilaments, and then where do you have all the thick, which means that you have all thick and some thin. So let's try that. Let's divide them up before we try to find them. So first thing I'm going to do is this only thin. So if I'm my only thin, I need to go this end of the thick myofilament, which is right here, and let's start dividing that. So from the Z line to this point, this first dashed line, that's where we're showing only thin myofilaments. I know that you're seeing this elastin, but let's just pretend it doesn't exist. Or this titin. Let's pretend that it doesn't exist for now. Because right now we're only looking at thick and thin myofilaments. So if you only have thin, this is your actin. This is your I band. How do you remember that? Once we get into this idea of troponin, tropomyosin, if that word overwhelms you, don't worry, we'll get to it. We'll come back and see how that helps you remember that this is only the thin. Okay, if we are going to do only thick, let's identify that. So only thick. So let's go to the end of my thin and draw a line down, because that would be only thick, and a line down, only thick. Well, if you notice, if I do that line as solid and this line as solid, what letter does this make? Maybe it'll be better if I do it in a different color, like yellow. So this and this. And I go across. What letter does that make? That's your H zone. So your only Thick myofilaments is H, your only thin is I, and what else does that leave? Let's do a different color. Let's do this green. If I'm going to do all my thick, then I need to go from here, the end of my thick, and I need to go over here, which is the end of my thick. This is called your A band. So I'm going to redraw this, and I'm going to start talking, or I'll talk through it as I'm drawing it. So the first thing I want to know are the ends of these sarcomeres called Z lines or Z discs. I know that I have thin microfilaments directly attached, and this is predominantly made up of the protein actin. I have these thick myofilaments here that are indirectly attached with these tightened elastic myofilaments. These thick myofilaments are made up of myosin. And if I want to separate some different things, then I can separate 3 different regions. I can do only the thin, so I go to where my thick starts, and then I'm going to go from there over to my Z line. If I only have thin, that's I. Then I need to do only thick, so that's where I took my yellow marker, and I did only thick. So this was thick, and this was thick. And I said this letter made the letter H, so this is my H zone. Then I know I needed to do all thick. So if I did all thick, let's do this. That'd be from, I need a different color. If I did from here, that's my thick, and that's thick. This is the A band. So we've got Z lines, we've got I bands, A bands, H zone, and then if you go straight through the middle, you can do an M line. So we can say this M line is in between the 2 Z discs. We can say it's in the middle of the A band. We can say it's in the middle of the H zone. All those things mean the same thing. Alright, so when the muscle contracts, we're going to get these thick myosin filaments pulling our thin myosin filament, or thin actin. We're just going to say thick and thin. We're going to get our thick myofilaments pulling our thin myofilaments, so these Z lines move from the Z line towards the M line. So we contract inwards. Alright, now we want to go into the structure of this myosin and actin. As you can see, we've gotten smaller and smaller through these pictures. So back here, we're looking at the whole muscle. Then we went into one muscle cell. So when we went into one muscle cell, we saw hundreds or thousands of these different myofibrils. And now we're looking at one specific part of the myofibril, or one individual sarcomere, and now we're breaking that up. And now we're going even deeper into the sarcomere, and we're going to look at the anatomy of this stuff. So we're getting really small here.